Good afternoon and welcome to the Todos Medical KOL webinar. At this time, all attendees are in a listen-only mode. A question and answer session will follow the formal presentations. If you would like to submit a question, you may do so at any time throughout the webinar by using the Q&A function at the bottom of your webcast player or by emailing your questions to questions@lifesciadvisors.com. As a reminder, this call is being recorded and a replay will be made available on the Todos Medical website following the conclusion of today's event. I would now like to turn the call over to your host, Gerald Commissiong, President and Chief Executive Officer of Todos Medical. Please go ahead, Gerald.
Thank you, and thank you for everyone for joining this call. The purpose of this call is to be discussing 3CL Sciences, which is a jointly owned company that is being formed between Todos Medical, 60%, and NLC Pharma, 40%, to advance the tremendous work around 3CL protease biology and 3CL protease inhibitors developed by Dr. Dorit Arad to address SARS-CoV-2 in a number of different ways. We've been working on this program at Todos with Dr. Arad since last year, and there are really two areas of focus. One, on the rapid and early detection of SARS-CoV-2 in a manner that is more efficient and earlier than antigen or PCR tests. And second, after we found some proof of concept in that area, we expanded to include dietary supplements and now botanical drug products. And that botanical drug product we call Tollovir is going to be the main purpose of today's conversation. Next slide. So moving into the executive summary. Next slide. Next slide. So just a little brief summary. Todos Medical is a New York headquartered but Israeli-founded medical diagnostics company developing blood tests for cancer and Alzheimer's. When COVID-19 happened, we were in the middle of an acquisition of a molecular testing lab called Provista Diagnostics, and we moved into the COVID testing business because of their PCR testing capabilities. However, the company's Videssa breast cancer blood test is intended to be launched in the first half of 2023, and that was really the focus of the company before COVID. When we went into COVID testing, we realized we needed to have something proprietary, and that's when we, through some advisors, connected with Dr. Arad and became associated and understanding of what 3CL protease biology. And so 3CL Sciences is this joint venture, and we're really looking to target the treatment and rapid detection of COVID. We have three products or product candidates that we're bringing forward in this area. One, which is going to be the primary focus of today, is botanical drugs, and that is Tollovir. This is an oral formulation of a combination of two strains of an FDA-authorized traditional Chinese medicine dietary supplement product that is now a botanical drug product that has within it a specific ingredient that was isolated, or a key ingredient isolated by Dr. Arad, that is a powerful inhibitor of the 3CL protease. But it also has some anti-cytokine activity, which we'll discuss later. Beyond that, we have dietary supplements that have certificate of free sale from the FDA, where we make no claims as it relates to treatment, but we were authorized, and the only known supplement that we know, authorized to have a 3CL protease inhibitor claim. So we have two products in this dietary supplement that have 3CL protease inhibitor claims. One is called Tollovir, maximum strength Tollovir, which Dr. Arad will touch on briefly with a little bit of data as compared with Tollovir. And then we have Tollovir Daily, which is a daily formulation, which again, Dr. Arad will touch on with respect to Tollovir and how it compares Tollovir, Tollovir, and Tollovir Daily. And then finally, as mentioned, we started with biomarkers, and that we call Tolo Test, and that is measuring 3CL protease biology in a upper respiratory tract swab to identify the presence of a coronavirus. And obviously in large circulation, we're looking for SARS-CoV-2. And we think that this could be for rapid detection, large scale replacement of antigen testing in a manner that's much more efficient and reportable. With that, we're really going to go now and focus. Next slide, please. On Tollovir. and Tollovir and its positioning with respect to other drug candidates, oral drug candidates for the treatment of COVID-19. So as we know, as we've looked at, there are really 4 candidates that we think are important in this area. We have a drug candidate called Tollovir. As mentioned, the mechanisms are 3CL protease inhibition and anti-cytokine. We're currently in a phase 2 clinical trial, and we have data pending. And provided the data is good, we expect to move into phase 2/3 studies in the hospitalized setting, and then expand into the non-hospitalized setting of severe critical, in moderate, severe. So that's once you get past the mild to moderate stage, but before you get to the hospital, and then looking at pediatrics because we're dealing with a natural product that's very, very safe. The competitors, obviously Pfizer also is late stage, and they have a 3CL protease inhibitor. This really validated our target, and we'll go into that in a moment. The challenge that they have is they do have an extender of the PK, which is ritonavir, but that is an important part of their formulation. So they do have strong data in the earlier stages of diseases, and they are pending FDA approval. We've been hearing that that may come as early as today. There's Merck, who also has an oral antiviral. That one is, the mechanism is not protease inhibition, it's mutagenesis. So a more extreme form of RDRP. So this is fourth mutation that is supposed to induce apoptosis. There have been concerns about potential variants emerging from this, cancer and birth defects, but there was some early data that suggested positive, and in the AdCom, there were some questions about whether that would persist, their positivity. And so the FDA is reviewing that now. We do expect a decision imminently alongside the Pfizer pill. And then finally, there's another later stage compound called Temple. That is being developed by Adamas. The antiviral mechanism is RDRP, so more similar to Merck than to our protease inhibitor or Pfizer's protease inhibitor. RDRP, we think, is somewhat weaker than 3CL protease inhibition, but it has shown some activity. And they also have an anti-cytokine component. So that's probably the most comparable, but we believe our mechanism, 3CL protease inhibition, is better than the RDRP mechanism. So they currently have phase 2/3 ongoing, and they will be reading out sometime in the first half of 2022. So those are the competitors in the oral antiviral space that we want to focus on. And when you look at the stage of disease here, this little chart on the left, it's really important to understand early on in the disease, it's really all about virus, viral replication. And then at some point, there's an innate immune response that's triggered, and then that can become a hyperimmune response. And that's really what drives the bus later stage in the disease. And so really managing this process from viral to inflammation, at the different stages, pre-hospital and hospital, that's really been the challenge with COVID. And I think some of the experts we have here on the phone today will talk about both the viral response and the inflammatory response. Next slide. So with that, I'm going to turn it over to Dr. Av-Gay. Please introduce yourself and talk about the antiviral mechanisms.
Hi, I'm next slide, please. I'm Professor Yossef Av-Gay. I'm from the Department of Microbiology and Immunology at the University of British Columbia. I hold a position in the Division of Infectious Diseases, Faculty of Medicine, and the medical school here in Vancouver. I run a research laboratory, a BCL-free research laboratory, where I do research on high sensitive pathogens such as SARS, COVID-2, TB, etc. I was involved in isolating the first SARS virus back in 2002. And I'll talk to you about the target of the 3CL protease that is a key compound of the SARS-CoV-2 biology and is essential for its replication. Some refer to it as NSP5 or MPRO or other names, but the main mechanism is related to a single protein that is a cysteine protease. Protease, by definition, is a protein that eats proteins and chops them. So the protease cleaves itself from the big polypeptide that I'll show you later in the slides. It cleaves coronavirus polyprotein at 11 concept size that are needed for the continuous biology and replication of a virus. And it was identified and validated as drug target to block many viruses, but coronaviruses infection specifically. And there is a proof of concept by Paxlovid by Pfizer, which shows that inhibition of this protease actually block the disease from progression, and they have a very good result in their clinical trial. Next slide, please. So the coronavirus is a human infection virus that came from the environment, came from animals, and its biology, it enters epithelial cells or tissue cells via a receptor called the ACE2 receptor. And as you can see in the slide here, it goes into the cells and empties its genetic material, which is called the viral RNA, the positive strand viral RNA. This positive strand goes to the biology of translation into a big polyprotein that encodes for proteins that are needed for next stages of a virus. This polyprotein has the free C protease built in it, and it cleaves itself as well as other proteins, which mediate the RNA replication and formation of a genomic DNA, which will serve as a template for transcription and making the particles of the virus that are needed to be encapsulated and bud out of the cells in the new cycle of the SARS-CoV infection 2. So in order to, there are some inhibitors that can block the cleavage of the ability of the SARS-CoV-2 CL2 to cleave the big protein. And these can be small molecules, some chemicals, peptide or peptide mimicking, like Pfizer has developed, which actually mimic the protein site that it was supposed to cleave. So then it fits the active site, or supposed to fit the active site, and blocks the binding of the polypeptide to the protein. But our focus is on plant extracts of what we call phytochemicals, which in nature are very prevalent. The SARS-CoV-2 is not the only virus that utilizes the free C protease for its biology. There are plant viruses that are utilizing the same system, and plants in evolution develop systems to overcome the infection. And these include plants chemicals or plant extract that can block and inhibit the formation of the contact between the free CL and its polypeptide. Next slide, please. So because of the ability of botanical extract to inhibit such a key biological effect of the free CL protease, there have been several clinical trials for botanical drugs in clinical trials. This is just a few days ago. There have been 6 clinical trials in addition to the clinical trial that Dorit is conducting in Israel. These clinical trials are directly aiming at humans. And if you see the next slide, please, are facilitated by the advantage of having a botanical designation for a drug at the FDA. The FDA drug approval pathway is a long pathway. Usually it takes between 8 to 20 years to discover, develop, go through preclinical development and through all the phases at the FDA that ended up in drug approval. However, for botanical, because of their intensive use and a large body of knowledge that shows no harm to patients, one can skip the early discovery and go directly after the IND and ethics to phase 2 clinical trials, phase 3, and it can even be used for emergency use authorization for botanical garden for hospitalized COVID-19, means that there could be even an enhanced and quicker approval of a botanical extract as a drug to treat specific diseases. This is not the first botanical. There have been botanical extracts that have been approved by the FDA, and I think this is the most suitable, efficient, and quicker way to the clinic, targeting the free CL protease. From here, I'll take it to David Greenberg, who will talk about the second activity.
Hello, my name is David Greenberg. I'm a pediatrician and I'm the chairman of pediatrics in the division in Soroka University Medical Center in Ben-Gurion University. I'm an infectious disease specialist and I was trained in Vancouver, Canada in BC. And today I will talk about the inflammatory response to the infection of the COVID-19. Next slide, please. So we know that there are different mechanisms how this inflammatory response that get out of control can happen, and this is called the cytokine storm. There are different etiologies that can cause this cytokine storm. These are some of the viruses, and the COVID-19, of course, is the most important one, but there are also bacteria that cause sepsis, and there are also other mechanisms such as autoimmune disease and others. And you can see on the right side what happened to a patient when this inflammatory response is getting out of control and there is overreaction of this inflammatory response in effect that the patient now has not an infection in the lung, but it has this inflammatory response that destroys the lung. For this reason, we have to talk a little bit about the immune system. Next slide, please. In this slide, you can see the immune system that has two arms. On the left side, these are the innate immunity, and these are the first responders that respond to any infection. And there are different cells that called the white blood cells that are responders. And these responders are increasing some mediators that call the cytokines. These cytokines are causing the inflammation. On the right side, these are the adaptive immunity that cause the antibodies. That this is what we do with the vaccines. When this system is getting out of control, there is more and more inflammation, and it's increasing itself in the fact that at the end, there is a destruction of some of the organs, such as the liver, the heart, the brain, of course, the lung, and the kidneys. At the end, it can cause a multi-organ failure and death. Next slide, please. I want to talk also about a different mechanism, which is not directed to the infection that we see in adult. In adult, when we see the infection, this cytokine storm can create after anything between 7 to 14 days. However, in children, we know that it takes a longer time, sometimes 3 weeks to 6 weeks, and then we have this inflammation as well. And this is a specific syndrome that called either a pediatric inflammatory multisystem syndrome, or PIMS, in Europe, and in the North America, it's called multisystem inflammatory syndrome in children, MIS-C. And this syndrome is actually very similar to what I just described, with increase in the inflammation that can also present as a multi-organ failure. I personally treated more than 40, almost 50 children with this syndrome that need to be hospitalized in intensive care unit because of respiratory failure, cardiac failure, brain damage and neurological problems, kidney insufficiency, etc. So this inflammatory response is also a late response that they create after a few weeks from the infections. Next slide, please. So why COVID-19 is different? First, because it involves more inflammatory cytokines than any other conditions that cause this cytokine storm, thereby providing an explanation for the aggressive nature of this COVID-19. Second, lymphopenia. This is the hallmark of the infection, and although relatively less frequent in other cytokine storm etiologies, it was often observed in patients with COVID-19, suggesting that COVID-19 may be a main attributor to the innate immunity rather than the adoptive immunity cells that I talked previously. And the third one is the compared to bacterial infection that induced the sepsis that I presented previously, the treatment of COVID-19 is more challenging because blocking the inflammation of the cytokine is not enough, because without effective antiviral drug support, it still can continue the infection and exacerbate the infection as well. So we have to normalize the immune response. And with that, I will finish and I will handle to Dr. Dorit Arad. Thank you.
Sorry. Hi, my name is Dr. Dorit Arad. I'm the CTO of NLC Pharma and 3CL Sciences. I'm here in the office with my partner, Avraham Marilus, in Tel Aviv. I've been working on the enzyme 3C protease throughout all my career, beginning in my postdoc study in UCSF, where I was introduced to the 3C by Peter Coleman, and afterwards in the university, Tel Aviv University, pharmaceutical companies and biotech companies. I was introduced to several aspects of the 3CL, and recently it was shown that 3CL has a very important role also in COVID. So I want to tell you about 3CL and about Tollovir from my perspective. Next slide. Next slide. Okay. So the 3CL protease is a viral enzyme. It is in the genome of the enzyme, and it is required for the replication of the virus and its transmission. It is conserved through all coronaviruses and also other viruses, as you'll see next. And for example, we started, bring it back, please, thanks. We started with the common cold virus. The 3C is a 34 kilodalton trypsin-like cysteine protease. It has a unique mechanism and has a unique cleavage target preferences. As Yossi described before, the RNA enters into the cell, and it has to be cleaved like scissors in several places in order for the virus to be a new virus, to complete its cycle and become a new virus. That's in the first line. If you inhibit the protease, I mean, you break the scissors actually, the virus cannot reproduce. So we are looking for inhibitors that will inhibit the reproduction cycle, and Tollovir has such properties, and we found it according to its mechanism, the mechanism of the 3C. So Tollovir is actually a mechanism-based designed preparation. Next slide. So, as I said, 3C and 3CL exist in several viruses, and they are trypsin-like cysteine proteases. They are, the main purpose is the reproduction of the virus. But we see that the 3C has other roles in the virus cycle. I mean, in the virus and in the disease. It is responsible also, for example, to the cleavage of a very important protein called NEMO, which is in the brain, but exists also in other places. And thereby, it turns off the cellular immune response and other alert mechanisms that control the immune system. So this may be a reason for turning on the cytokine storm. There are other factors that turn off some factors in the immune system, in the immune system, and they also could turn on the cytokine storm. The main thing is that the cytokine, the Tollovir, has a dual action. It targets both mechanisms. It targets the replication, and it targets also the reduction of the cytokine storm. Next slide. So the Tollovir is a botanical drug. We got it as an extract from a natural compound, a root of a natural compound, because we knew that it contains some active material, which we knew that it is a 3C protease inhibitor. But we got many surprises on the way when we started to isolate the materials inside the root. We saw that some isolates really inhibit the 3C protease, and they have antiviral activity in viral replication. But there are some isolates that inhibit the cytokine storm. So another function of this botanical potential botanical drug is to normalize the immune function. So actually the Tollovir has a synergistic activity. It has key ingredients that allow us to treat patients even in the hospital, even hospitalized patients. Next slide. So before we turn to the results on the hospitalized patients and the clinical trials that we are doing now, I want to say a word about the comparison to Pfizer. Pfizer is working on the same target. The target is the 3CL protease. What Pfizer claims is that they inhibit the replication of the virus. Pfizer is a structure-based design compound. It is actually a peptidomimetic compound. It is a peptidomimetic compound, and ours is designed by a mechanism. So the difference between mechanism-based design compound and structure-based design compound is that the mechanism-based goes directly into the active site, whereas the structure-based is dependent upon the binding sites in the enzyme. Let's go for a moment to the Omicron mutation. In the Omicron mutation, there is one mutation in the 3C in the binding site. This mutation did not hurt the Pfizer inhibitor, the Pfizer drug, but we don't know what will happen in the next generations. We think that our Tollovir will stand up even if there will be other mutations. Another point is the activity. So we see in the graph that Pfizer and Tollovir, they reach finally a similar activity. The difference is that Pfizer needs like 1 or 2 capsules, and we need 12 capsules to reach, to inhibit the enzymes. We hope that in the next generation, we'll be able to optimize the compound and reduce the dose. But currently, it's sufficient because it does the work. I mean, we know that it inhibits the protease, and we get the patients are, we help the patients. Additional thing is that Pfizer has a additional compound which is called ritonavir, and this is to keep the pharmacokinetics, while we have a compound which does not need the help in the pharmacokinetics. So it may create an advantage, for example, in giving it to children, or even starting the medicine or the natural compound earlier in the course of the disease. Now, much beyond the inhibition that we mentioned before, we discovered that the Tollovir, as I mentioned before also, has dual action. It is not directed only to the replication, but it has also a role in stopping the cytokine storm. And because of this, it may help hospitalized patients as well. So next slide. We did an observational clinical trial in the beginning of the corona. It was a very small trial in which 10 hospitalized patients got the Tollovir, and 17 didn't get any treatment. They got the regular treatment that the patient got when they were hospitalized with the disease. So the results were quite amazing. The results were that all the 10 people in our group, they just went home after a regular course of events, like 5 to 6 days. And the amazing thing was that the CRP, the inflammation factor, was reduced dramatically. In the other group, at the same time, and in the same hospital, like 5 people died from the disease, 5 and other 5 were incubated, and the rest, the CRP got down very slow, and the course of the disease was not normal. I mean normal because they in the end they recovered, but slower. So based on these studies, we have also examples that in which we gave some ingredients of the Tollovir in a supplement that were in the supplement called Tollovir. We gave it to several people in the US and in England, and we had stories which were amazing. So based on this, on all the data and the fact that we got activity in the lab, we decided to perform a double-blinded placebo-controlled trial in Sha'arei Tzedek, which was going on for like 9 months. And we are starting the analysis now. And Gerald will speak about it.
All right, thank you very much, Dorit. Next slide. So just, because I know we saw the information and I've had many questions, when you see those graphs of comparing our compound with a Pfizer compound and the activity, just so everyone understands that, the left slide, which was the one showed identical activity, that was really the Tollovir. That middle blue line, that was more of our Tollovir dietary supplement product. So as part of the release criteria, the data we submitted to the FDA, that's why we were allowed to get the claim of 3-cell protease inhibition for the maximum strength Tollovir. And then the slide on the right that showed obviously much less, that's really the Tollovir daily. So, that is the difference in activity that, when people are asking with respect to Tollovir daily, Tollovir, and then ultimately Tollovir, which we're talking about here, that has in addition to the protease inhibition activity of Tollovir, has some additional high concentration anti-cytokine extract that we added to the mixture to optimize and really bring out additional anti-cytokine activity. And that is why we decided to go into the hospital, because we felt obviously that Pfizer was going to be a major player early on in the disease and we would have challenges. We have good supply, but obviously with a natural product, it's not infinite. And that's another reason we decided to go later in the course of disease, where there was less competition and where the dual mechanism made sense. Because, we think that we can actually serve that later stage population in the hospital and the pre-hospital, but yet pretty sick population. We can serve them as well to keep them out of the hospital. And from a pharmacoeconomic standpoint, we think those are really extremely valuable use cases, reduction in time in the hospital, and then obviously not going to the hospital at all, and which also reduces risks, of many, many other aspects of being hospitalized. So in the current phase 2 trial that we have, where we have 31 patients enrolled, 1-to-1 randomized treatment to placebo, the primary endpoint is time to stable disease. After several discussions with Dr. Jules Mitchell, we looked to optimize everything based upon the current thinking, and that really is time to stable disease. 2 key endpoints, secondary endpoints on the clinical side, are reduction of time in the hospital, of course, and death. We saw both of those in the observational study, and we think that we can hopefully, with the next set of data, we can replicate that observational study, which will really give solid proof of concept to the work that we've done, throughout the last year and a half, with regards to optimizing dosing, figuring out treatment regimen, number of pills, and then ultimately getting to a clinical outcome. And then obviously the next thing, which has become increasingly important over the last 6 months, especially for regulators, are those biomarkers. As you saw what Dr. Arad said, we saw CRP reduction in the previous study. We also, from our clinical work in helping patients, have seen impact on D-dimer, which is blood clotting, also IL-6, as well as a litany of additional cytokine-related markers, including galectin-8 and others, that we will be adding beyond these markers. So we think that the biomarker story here and profile could have a profound impact, especially because of some things that Dr. Arad has been working on that will soon be published as it relates to normalizing the innate immune system. Next slide. So in terms of the regulatory pathway for investors trying to understand where we are, so we are going down the botanical drug pathway. So obviously the safety is key, and that's why botanical drugs have an expedited pathway per Dr. Av-Gay's statements. So we think, given that we have a certificate of free sale for some of the ingredients, but we've optimized the mixture with Tollovir, and we have very strong quality control. So we can get consistency, and we've spent a lot of time qualifying supply. and putting things together,
Yeah.
we think that we can absolutely bring something forward. So our primary clinical development plan, of course, is hospitalized to complete this phase 2 readout. We intend to request EUA in certain jurisdictions where we've already received some preliminary engagement of interest. Obviously in Israel, where they're actually seeing this live in patients. And we also have a partner in Greece, where they are doing some work there to engage with regulators. We are looking at other pathways beyond just those 2 jurisdictions. So we've retained a consultant in Germany who's identified certain pathways for a botanical drug product, and we will be filing an IND in the US and engaging to determine the FDA's posture and what, if any, additional data will be required. We're obviously expecting that we will have to conduct additional studies with or without an initial EUA. And so we're preparing for phase 2-3 trials in hospitalized patients, and then also looking resources dependent, of course, on expanding into other areas, secondary, moderate. We're really calling that, pre-hospital. So you can be pretty sick, moderate, but as long as you're not in the hospital, we're calling that moderate. That's one area we think that could have a big impact. Pediatric, as Dr. Greenberg mentioned, and Dr. Arad mentioned, that there's nothing right now for pediatric, and people are very scared to bring new products into the pediatric market for safety concern issues, especially given that children in general do well. We obviously, as a natural product, and given the safety we've seen, including safety to date in the dietary supplement that children have taken, we've not seen any toxicity. So that's very positive to date. Obviously, we're going to continue to monitor that closely, but we have not seen any. And so we think we could have potential in the pediatric market. And then finally, some of the aspects, and this is really new biology that we've been discovering with Dr. Arad, as mentioned, this innate immune system and the downregulation and impact on the innate immune system, we believe is driving the hyperinflammation, which we believe is what drives long COVID. We think that there's some very commonalities between the different aspects of long COVID in terms of endothelial cell oxygen transfer, and then it's just about the weakest point in the chain. That's where you end up having the problems. So we think if we can help normalize that function over time, we could help improve that mechanism and ultimately have an impact on long COVID. So we are working to identify really the places within long COVID, the subcategories we want to go first, where we think we could have the biggest impact. Next slide. So in summary, what's the background of Tollovir? It has significant prior human safety data as a botanical drug candidate, and obviously the ingredients as dietary supplement. With the supplement, we make no claims to diagnose, treat, or prevent or cure any disease. Of course, it's just 3CL protease mechanistic claims. We hope it helps people, during this challenging time. This Tollovir has potent anti-3CL protease inhibiting compounds within it. So it's not, some mixture that synergistically together inhibits the protease. We've identified, Dr. Arad has identified within the mixture the compounds that are driving the bus with respect to the 3CL protease inhibition. There are also key anti-cytokine compounds. So these are our NLC extract 1 and NLC extract 2 that was identified by Dr. Arad earlier in SARS-CoV-2 that really help with the hyperinflammation. And it's important to note, this didn't just happen. There's been $20 million invested in this since 2002, $5 million by us at Todos in helping NLC create this new set of data. So there's really robust experience and investment that went in to bring us to this time. We have a phase 2 randomized double-blind placebo-controlled and hospitalized patients, 31 patients enrolled. The data analyses are pending. We do have something similar to a DSMB called the Trial Level Safety Review that Dr. Mitchell helped us put together. A data lock is expected very soon, with data reading out hopefully very early in January. Most likely, could be late December, but most likely it'll be early January. So we want to make sure we provide timelines to investors. So those are the timelines. So not long, but we can't say it's going to be tomorrow. We've got, there's a tremendous need for a dual action, as Dr. Greenberg said, and we think that we can fill that need. And we will be preparing for the phase 2-3 that we expect to start, obviously in the first quarter, because we think this is an emergency and we can get expedited review in a number of jurisdictions. So it won't only be the US, but ex-US as well. And we'll be filing an IND in Q1 2022 after this data, and engaging with regulators both in the US and Europe and worldwide. So with that, this is the presentation on Tollovir, and I will open up the floor to questions.
Thank you, Gerald. At this time, we'll begin conducting our Q&A session. As a reminder to the audience, you may submit your questions through the Q&A function at the bottom of the webcast player or by emailing your questions to questions@LifeSci Advisors. It looks like we have one question coming in live from Michael Irwin at US Invest. Michael, you may go ahead and unmute your line.
Hi, it's Michael Irwin from Univest Securities. I have a question regarding the pediatric side. As you mentioned, it does take longer for the hyperinflation to be found in these kind of patients for 3 to 6 weeks compared to 7 to 14 days. So would Tollovir more so be a long-term kind of treatment because it does appear in the later stages and especially further out post-infection?
So I'll speak from our development standpoint, and then maybe Dr. Greenberg can just chime in with regards to the biological aspects. We haven't identified the dosing schedule yet, and obviously we don't think it'll be anywhere near as many pills for the kids. But we do know, yes, this can absolutely be taken over a long period of time. I personally, have been taking these ingredients for 18 months. I know, my kids have been taking them for, quite a long time as well. So, we don't see any issues with respect to chronic exposure, if that's the question. From a biological perspective, Dr. Greenberg, maybe you could chime in on what you think the dosing schedule would kind of look like in terms of period of time.
So as you've said previously, there are 3 different stages and 3 different products. So one is for the maybe prevention, the other one is to start to deal with the infection, and the third one is to deal with the inflammation. So if we do know, one of the potential effects that we can use is when we do know that the patient is infected, we also can start to prevent the inflammation. So at the beginning of the infection, we start to prevent the inflammation, and hopefully at the end, we are not going to end with this inflammatory response, which calls the MIS-C, and reduce this. And this is definitely should be one of the endpoints of the studies, of course.
Okay, so if you treat it at the beginning, you shouldn't see these kind of hyperinflation found in children and other patients.
Absolutely.
Okay, thank you.
And in fact, in responding to that comment, that might be actually one of the reasons to even though kids are not very symptomatic, to intervene. Because we do know, even if you're not very symptomatic, you can still lead to long COVID. We think because of the endothelial cell damage that persists over time. So if you have a way of stopping the 3C and kind of stopping that damage, you could have potentially effects down the line. That's one of the reasons, even if you're not very symptomatic, to treat, because there are other things other than just hospitalization and death to deal with COVID.
Okay.
And I can add something that from our experience on children, especially Avraham Marilus, children react very fast to the medicine. Actually, when we started, there was one baby that got the drug through his mother breastfeed. And he got better immediately.
Thank you, Dorit, and thank you for the questions, Michael. I'll now turn it back to Gerald for questions that came in over the webcast.
Okay. So with regards to questions from the webcast, can we get enough supply to the marketplace? The answer is yes. We feel we can get sufficient supply to the marketplace. Because we've been working on that for quite some time. So as I mentioned, we've qualified, or Dorit and her team have qualified in excess of 40 different manufacturers. And as a result of that, we've got a very strong understanding that we can do this. We are, in fact, also looking at our own manufacturing ourselves in acquiring a facility and all of the raw materials, so that we can grow them naturally ourselves. And we've got a very strong plan for that. So for us, it's really resources dependent. We want to make sure we create value for our investors at every step. And then when that value has been created, we monetize properly and diligently so that we can take it to the next step. And one of the next things we're going to do after we get this data, obviously, and prove, hopefully, of course, prove that our thesis is correct, we will then look to really gear up and maximize that plan. But right now, obviously, we're just manufacturing Tollovir and getting that out to the market. So what I see from Yi Chen from H.C. Wainwright, what is the revenue you have been generating with sales of Tollovir? What is the gross margin? I won't get into the gross margins because we are negotiating with a number of groups. From a revenue perspective, it had been pretty light until recently. So, $2,000, $3,000, $5,000, $10,000 a day. But recently, in the last couple of weeks, we've hit up to $100,000 a day in sales, and we're starting to normalize, we think, somewhere in the 30 to 50,000. And we have not done any marketing. We actually just hired a head of marketing last week or a couple of weeks ago, but we just announced him early this week. And now we're going to be pushing some money towards marketing. So, for us, we're resource constrained, and so we are managing that. But we do think that there's very significant opportunity to market the Tollovir dietary supplement because that's available now. We can't make any claims with respect to treating COVID, of course, but we do think people understand the benefit of having 3-cell protease in their system if they encounter, as well as if they've got more significant issues to take the maximum strength. So again, we can't get into the use of the supplement in treating any disease, but we do think that people understand the value. Pfizer's antiviral was just approved by the US and needs to be prescribed by a doctor after testing positive, which we know may be hard given the number of days. If your results come out as positive and get an EUA, will it be easier to get sick patients your antiviral in the hospital? Yes, of course. So, once you get into the hospital, everything changes with regards to access. So we don't anticipate, if the drug works and it's authorized and physicians see the results that that we're expecting, then we don't think it's going to be a challenge to get it to those patients in the hospital. Again, because it's a smaller number of patients, we can ramp up production to meet that capacity, we think, pretty easily. In terms of the prescription for the earlier stage, that's going to be a big problem. I think everybody in the whole country recognizes the challenges with testing now. Everybody sees the lines, especially in New York. It's not clear whether the FDA is going to require a rapid test performed by a healthcare provider or a lab to be positive, or if they'll accept a home test to get access to the drug. It's still unclear. Obviously, a PCR test, we're having a lot of problems with turnaround time now. We've seen some labs actually get in trouble with that in terms of giving results. So the window in which that drug can be prescribed practically is shrinking when there's spikes. And so, you kind of end up in the situation where unless we really expand our testing infrastructure and make it easier to test, then you're going to have challenges with getting Pfizer's compound out there to the masses early, and then it's going to have less efficacy and there'll be more patients later on. So it's just something we're going to have to deal with. But we do think we have an advantage because our window is much bigger, window of treatment, and that's really what we're targeting. So you had a press release today about a partnership for testing. Do you think this partnership for Provista will bring you to full capacity of 20,000 per day? No, we don't think that will get us to 20,000 per day alone. We do have a number of different products coming, but that's not really Tollovir related, but thank you for the question. Let me go through the next set of questions. Can Tollovir be used prophylactically? I can't answer because with regards to prevention of any disease, because we can't make that claim. What I can say is we do think it makes a lot of sense to have a 3-cell protease inhibitor in your system if you do come in contact with COVID-19. That we can say, because then it can start its action early, obviously. And you know, there are a couple of factors. How much virus you take in, you know, is a big factor with, you know, the amount of virus you're ultimately going to have. Because if you're taking a small amount, it starts to replicate from a smaller base. Omicron seems to replicate quite well. So that may not matter after a period of time, but if you start to cut down its ability to replicate earlier, then you provide your immune system time to catch up. And that's really, you know, the objective of having a 3CL protease inhibitor in your system, is to be able to slow down the replication to allow it to catch up. So I will go. Apologies. I did have a question with regards to Tolo test. Will the Tolo test be available as an at-home test? What is the anticipated schedule for approval? So just touching on that briefly, right now the focus is not on the at-home, only because there are just a number of challenges from a regulatory perspective. The oversight is tremendous, and we think that's why there's so few at-home tests approved. Tests that are kind of self-contained, that are done at the point of care, where there's some oversight, those seem to be much more available. And so that's going to be the first regulatory pathway. We are targeting for large scale testing. So we're thinking airports, schools, office buildings. That is the focus. And it's going to be at the point of care in a machine and automated to be able to provide fast results electronically, so that, you know, people can get access or not. It's really a choke point kind of test that we're focused on. One question from John. Is the 3CL protease inhibitor the end all and be all of solving COVID, or will COVID require other unknown treatments to slow it down? Right now, you know, based upon the data we've seen to date with the current known variants, including Omicron, it looks like the 3CL protease has great activity in inhibiting replication. You know, that is clear. Doesn't really look to be impacted in any way by which variant you have. Obviously, the sooner you have it in your system, the better. That we can say, but independent of which variant, it helps to slow down, and it helps these other aspects, Nemo and others that weren't originally thought of, but are appearing to be coming important with relates to the immune system in COVID. So that we can say. We do think over time, this virus could evolve, and because we obviously no one has any idea what's going to happen, and it may, you know, there may come a strain at some point that, you know, you can add to the 3CL protease that could ultimately help it. Right now, it doesn't look like that's required, but, you know, like everything else in COVID, what we know today could be, you know, effectively useless 3 months from now, or 6 months from now, or a year from now. But we do have a high degree of confidence that no matter what cocktail ultimately emerges down the line, that the 3CL protease is a canonical base of that cocktail. And so we think that 3CL protease inhibitors are going to be very, very important for a long period of time with respect to COVID-19. So, all right, I only have 1 more minute here for this. Is there a limit on the time between the onset of symptoms and the taking of Tollovir by a COVID-19 hospital patient? And if so, is that a concern? It doesn't appear to be a timeline. So we have treated patients, including in this trial, that are intubated. So, you know, it's pretty much as late stage as you can get. We did that. Obviously, this is an oral pill, but if you take the pill apart and you dilute the pill, you know, into liquid, it can be taken, you know, as a liquid. I myself have done that, you know, plenty of times. If I thought I may have come in contact, just to coat my my mucosal wall. So we believe that there is no limit. You know, there's no too late to help calm down the cytokine storm. So, you know, with regards to that, we have not really seen anything else that has this dual mechanism that could do that. There obviously are plenty of people going after cytokine storm. You know, we know Humanigen is going after it. Cytidine is going after that. And so but as a dual mechanism, including an antiviral, no, we don't see any kind of late limit on that. With that, I think we don't have any more time for questions, but I thank everyone, and we'll try to answer as many of these as possible afterwards.
Excellent. Thank you, Gerald. This concludes today's event. You may now go ahead and disconnect your line.